Good morning, everyone. Today I'm going to be talking to you about a recently emerged virus that spawned epidemics of international concern. One whose rapid spread brought fear and panic, while the virus and associated disease remained a black box of the unknown, with researchers and public health professionals scrambling to catch up. While this description may sound eerily familiar, I'm not actually talking about the elephant in the room, but rather Zika virus, which followed its own meteoric rise into the public consciousness in 2016. Even though those epidemics in South and Central America have since burnt out, Zika and other flaviviruses, the family of viruses that Zika belongs to, still infected over 400 million people last year alone. Compounding this issue is the lack of effective vaccines or antiviral treatment options for Zika and many other flaviviruses. To rectify this problem, it's necessary to gain a better understanding of how Zika and other flaviviruses function at a molecular level, so that we can identify new options for antiviral treatment development. Like all their viruses, Zika needs to synthesize new copies of its genome, a process known as genome replication, in order to complete its viral life cycle and produce new viral particles that can infect neighboring cells. As a positive sense single-stranded RNA virus, Zika accomplishes this goal by first making a negative sense RNA copy that is complementary to the original positive genomic strand. This intermediate can then be used as a template to then synthesize new copies of the positive genomic strand, which can then be incorporated into newly assembling viral particles. We know that there are structures at the ends of the positive strand that are involved in recruiting the viral polymerase, the machinery that synthesizes the new strands. But until now, the structure of the negative strand intermediate has remained completely unknown. In my project, we've identified structures at the end of the negative strand that are conserved across several flaviviruses. We believe that these structures are involved in recruiting the viral polymerase and initiating transcription from the negative strand intermediate, acting as a promoter for synthesis of new positive genomic strands. By introducing mutations that either disrupt or rescue formation of these identified structures, we seek to answer three important questions. Firstly, what are the minimum structural requirements for recruitment of the viral polymerase to the negative strand intermediate? Secondly, how do these structures actually recruit the polymerase? And finally, and most importantly, based upon this information, can we actually inhibit this interaction? Answering these questions will allow us to shed light on a dark corner of the Zika virus life cycle, with the hope that such insight will lead to new targets and strategies for antiviral treatment development, so that when the next viral threat emerges, maybe we won't be caught so flat-footed. Thank you.